it's my pleasure to introduce our speaker today for the COVI Lecture Series. It's my friend and colleague, Dr. Jeffrey Stuart. Jeff is one of our CCOVI Fellows and also an Associate Professor of Biological Sciences here at Brock University. He holds a Bachelor of Science in Biology from the University of Guelph, and then went on to do his PhD in Zoology, also at the University of Guelph. He then went for a little time away from Canada to do his postdoctoral work in biochemistry at Cambridge University and in the laboratory of molecular gerontology at the National Institute of Health. He then returned to us here at Brock and currently teaches human physiology, cell physiology, and the biology of aging. His research focuses on molecular mechanisms underlying longevity and identification of small molecule modulators of pro-longevity genes. That's a tongue twister for me, Jeff. So today he's going to be talking about investigating resveratrol's anti-cancer and neuroprotective effects, but I thought it's quite interesting. He's also looking at this from a red wine metabolite perspective, so he's got another title to share with the group today. So
yes.
With that, Jeff, I'll leave it to you.
Thank you, Debbie, and thanks everybody for coming. And yes, I did change my title. The title is one that I didn't actually make up. It comes from a marketing campaign for around the graduate student whose work I'm going to mainly present. And she was actually Brock's first recipient of the Vanier Scholarship, NSERC's Vanier Scholarship. So the marketing campaign was about that. Decoding red wine. At the time we thought that was funny marketing speak. Subsequently we realized that it's actually quite appropriate for what Ellen has been doing, and you'll see as I go through this why that's the case. I'll just start by going a little bit off the path that I'm going to go down and tell you what the lab does, what my research program is all about. And I can divide it roughly into 4 different categories. I'm really interested in the biology of longevity, not so much aging maybe, but in longevity, what makes things live longer, things that naturally live longer, what are they doing differently? So we do a lot of work with many different species. We keep banks of tissues and cells from about 40 different vertebrate species at this point, and we're querying those samples to identify genes that are expressed at higher levels or sometimes lower levels. We find things to do with cellular stress resistance, which are related to resistance to neurodegenerative diseases, for example. We find things that suppress the formation and growth of tumors are really important. When you're long-lived, you've got to do those things. When we do identify genes that are important, what we'd like to be able to do is find ways to actually turn them on or turn them up. And that's really what the talk today is going to focus on, is that part of things. Something I will not talk about is we do work with some anti-cell death molecules, some anti-apoptotic molecules that Jeffrey Atkinson at Brock produces. But today I'm going to talk about some of the anti-cancer activities of molecules that Vitis vinifera produce, so the grapevines produce, and you'll find them in the leaves and also in the grapes themselves. So this is Ellen Robb, and what I'm going to talk to you about today is really all of her work, her PhD thesis, which is coming to, the work is coming to an end, and the thesis is being written now. Okay, so this is the starting point for this talk, is the idea of phytoalexins. And phytoalexins are molecules that all plants make, including Vitis vinifera, the grape, that all plants make under conditions of stress. So quite a variety of conditions of stress will lead to the production of these phytoalexins. One kind of stress that is relevant is fungal infection. The phytoalexins, many of them are based on, especially in Vitis, are based on resveratrol, this molecule resveratrol, which you're going to hear a lot about in the next few minutes. And these other molecules, pterostilbene, pisaids, there's a bunch of resveratrol oligomers. They're actually all based on the central structure of resveratrol. Small modifications, sometimes oligomerization. Those phytoalexins are produced to stop the fungi from growing. So things like the downy mildew. And perhaps they do other things as well, but they are very good at slowing replicative growth of fungi. And in fact, they're also very good at slowing the replicative growth of human cells, any mammalian cell. And that is an interesting property, and that is something that we've really tried to hone in on and understand. Just briefly, I'll tell you a little bit about phytoalexins then. Here's a table I took from someone else's paper showing the production of these, so showing these molecules turning up on and being measurable on the leaves of grapevines. And what we have here is actually 0 days, and then 2 and 6 days post-infection. So DPI is days post-infection. So an intentional inoculation of the plants with this fungi, and then measuring the plant's production of these molecules. And what you'll see is they are there. So resveratrol and some of the other molecules are there at day 0. Here they're not detectable. So they're there, but they're very low, at very low levels. In this study, below detectable limits. What's interesting is just that by 6 days post-infection, you do get these quite high levels of all of these things being produced. If you were to take samples from those leaves, and the same thing is true on grapes, that study happens to come from leaves, but you would see something similar with on the skin of grapes. You could actually extract all of that material, all of those molecules, and isolate the individual components. And that's the Decoding red wine title really comes from this idea. Those will end up in red wine if they've been produced in the grape, and then that grape is being fermented into a wine. They're going to be extracted. Most of these are highly hydrophobic molecules. They like to be in an ethanol solvent, so they'll come out into that wine as it's forming. So whether you were to do this in grapes or leaves or wine, you could actually separate out the individual components. And so these numbers each represents one of the kinds of molecules that were on the table preceding. Now, it turns out that probably all kinds of these have biological activities. And that's what we're interested in characterizing, is what are those biological activities. What we know quite a bit about is resveratrol, which is one of the peaks. So if we go back, just a single one of these peaks would represent resveratrol. The other peaks are different things, but the other peaks are all in Vitis. They're generally molecules that bear strong structural relatedness to resveratrol. So they are not resveratrol, but they look a lot like that molecule. Now, some of them, I'm showing here some oligomers, and the reason to show them is just to highlight that the oligomers of resveratrol, which means a number of molecules of resveratrol coming together and being built into a single molecule. These are reactions that are catalyzed by the grapevines themselves. These have really not been characterized. They're very difficult. They're somewhat difficult to isolate. They are not available commercially at any reasonable cost, and so they're things that we have not yet really been able to characterize, but would like to. What we have been able to do, what we can get and work on under reasonable conditions, is something like resveratrol. And some of the other single molecule versions of resveratrol, Pisaids and pterostilbene. And that's the resveratrol has become a famous molecule. It started to become famous in the late 1990s. People identified that as being something that was related to the French paradox, the resistance to cardiovascular disease in the French population, despite the consumption of high-fat diets and that kind of thing. But subsequently, resveratrol has been tied to neuroprotection, so protection against brain injury and neurodegenerative diseases. Some anti-cancer properties, all kinds of properties have actually been attributed to resveratrol. So there's a lot of interest there. And really what we want to know is how it works, what are the mechanisms. And as you'll see as I go, knowing the mechanisms, knowing how resveratrol can actually have all of these different effects that seem unrelated. An anti-cancer effect, an anti-neurodegeneration effect, some cardiovascular things. They do seem very much different things going on. And so what we're looking for is some kind of central mechanism that underlies a lot of different observations. So Ellen Robb's work really has been, it started out as entirely just trying to understand the mechanisms. You'll see that it expanded from that after we think we know, pretty sure we know the mechanisms. We were able to find like molecules like resveratrol that can elicit the same kinds of responses in animal cells. So this is one of the earliest experiments she did. And what she did was to take human cells growing in a dish and expose them to resveratrol and to measure a whole bunch of things. So we didn't really know what we were looking for. Well, we had some ideas, but we didn't know what the answer was going to be. This turned out to be the answer. Now, I say that because a lot of things didn't change. Many of the things that we measured, which I won't mention at all, didn't change, and we had this one enzyme leap out at us as something that was really remarkably changing. And the change was an increase. It was being induced. It was being induced by close to tenfold. So this was the expression of this one kind of enzyme by increasing by about tenfold in cells exposed to this resveratrol. What is the enzyme? It's a mitochondrial superoxide dismutase, manganese sod. I'm going to shorten it to manganese sod for the duration of the talk. I'll tell you a little bit more about what it does in a few seconds. But for now, I'm just going to highlight what we found. So manganese sod, this huge induction that was elicited by exposure to resveratrol in these human cells. And that really caught our interest. It caught our interest because I already knew about manganese sod. Based on my background working with mitochondria and working with reactive oxygen species, manganese superoxide dismutase is an antioxidant enzyme. And it catalyzes just one reaction, which is the dismutation of superoxide to hydrogen peroxide. These are two molecules that we don't need to know a lot about to appreciate the results right now. So what we wanted to know is that just a result, an isolated result that's peculiar to that one type of cell, those were epithelial cells, those were fibroblasts, or was it something that was more generally the case, something that happened to all cells? All cells respond to resveratrol this way. So it's easy to do the experiment in other types of human cells, and when we did those, we found that indeed the same kind of effect was elicited in brain cells and lung cells and lots of different types of cells. We did lots of them in species that aren't, in non-human species, because we do a lot of work in other species, other mammals in particular. And we found that it broadly will induce this manganese sod and lots of other species too. Now, being able to do that in cells in a dish is important, but ultimately, if this is something that people are going to take, and of course you can take resveratrol, you can go to a number of stores. You can go to even Zehrs probably now and buy resveratrol as a nutritional supplement. And if you're going to do that, what we need to know is, resveratrol might have this effect in cells growing in a dish. What does it do in vivo? And as a starting point, we did some experiments with mice. So we took mice and we gave them their normal diet. And we also gave them a high-fat diet, a diet that would make them fat, lots of caloric content there. And we put resveratrol in that diet, or we didn't put it in the diet. And we had another way that I won't talk about at all, which is a mini pump. It's a surgically implanted device that delivers resveratrol continually to them. So we had 3 ways to deliver resveratrol. And what we found is that we could, and it depended on how we gave the resveratrol in the diet, but if we packaged it into this vehicle of fat, then it actually must be getting to the cells, which we know because we can duplicate the result. So especially in brain, it seems to accumulate in a few parts of the body, including brain tissue, and you see this same increase, which is fairly substantial, about a doubling of that important enzyme, manganese sod. Okay, so now finally I'm going to tell you what manganese sod actually does. Now, probably most famous as an antioxidant enzyme, and the whole idea of antioxidants. So even resveratrol itself is an antioxidant. It's a chemical antioxidant. But I'm going to deflect your attention away from that idea that antioxidant is good, and that that's all of the effects that we see are because of this chemical antioxidant property. We don't think it matters that resveratrol is an antioxidant. We don't think that some kind of broad antioxidant capacity is even important. We actually think that the result, that the effect is generated much more specifically at a much more specific level, and I'll explain that. Here's manganese sod. It's found in mitochondria of all cells. It's an essential enzyme, and it catalyzes just that one reaction, superoxide to hydrogen peroxide. Superoxide and hydrogen peroxide are reactive oxygen species, so they can damage macromolecules. They can certainly damage DNA, proteins, lipids. And that would be true at very high levels. It's not true necessarily at normal physiological levels. So what is definitely the case is that at normal levels of superoxide, hydrogen peroxide, and similar molecules, these actually have important signaling functions. So we need to interpret what's going on there not as some overall damage response, but actually as a more fine-tuned response where the reactive oxygen species are actually chemically modifying probably specific proteins. So we think specific proteins. That's being shown here a little bit. Here's hydrogen peroxide, the product of that enzyme, and a whole series of reactions that take place involving proteins as substrates. So the substrate protein is a non-oxidized version, and the product protein is oxidized. In other words, you've made a small chemical modification on one amino acid. Turns out that that's really important and can completely change what the protein does. Now, what we know, and I won't show you this, but what we know is that manganese sod, by virtue of this reaction, we're not exactly sure. We're doing some work in this area, but we don't know exactly what's happening. But we know that the proteins that are being changed are cell cycle regulating proteins. So what are those proteins? Those are the things that actually coordinate cells moving through the cell cycle. In other words, during which time they're making new DNA, replicating their DNA, and dividing and becoming two cells, and those cells doing the same thing. That's what's being regulated by manganese sod. That was already known. That was something somebody else did, and they did it cleanly, using transgenic overexpression and also underexpression, knockout, and they had shown all of that. What we didn't know is that these things that red wine produces, the wine plant produces, can actually elicit, are very good at changing manganese sod levels. So what happens is when the levels of that enzyme are really low, we see very high proliferative rates. So cells divide very quickly. Now that's a clean experiment, an experiment that's done where we can go in and just actually play with the gene and cause it to make less of that protein. So we don't put anything else in, we just focus on that. That's the only thing that's required to actually make cells grow really quickly. You knock that down, that level, and cells start to take off. On the other hand, if you were to go in and do the opposite, put more of the enzyme in, no resveratrol, no anything else involved in this experiment, they slow right down. So we think that that's actually a really important effect. And we know that that's what resveratrol is actually modulating here. So here's the effect of resveratrol. We've got two data points here, there and there. On the y-axis we have average population doubling time. Now the higher number means it takes the population of cells longer to double. In other words, more time for cells to divide. And you have to think about that in the opposite way. That means that they're dividing more slowly. So a higher number actually means slower growth. That's on the y-axis. The x-axis is manganese sod levels. Now we've standardized it to mitochondrial number there. We'll ignore that too. So we're looking at the level of the enzyme, manganese sod, versus how fast the cells grow. And the two data points are a control, so an untreated cell, a bunch of untreated cells, and resveratrol treated cells. So this, if we start with the control, it gets deflected up and to the right. To the right means we've got a lot more manganese sod produced, and the rate at which the cells are dividing has slowed down. Those are measurable, reproducible effects in all kinds of different cell lines. So that's what resveratrol is doing. Now, the fact that it can increase manganese sod and also slow cells growth down really is not at all causative. Of course, we suspect it is because we know that if we were to do that without resveratrol, leave that out, and just target manganese sod directly, we can reproduce the result. But we still have to show that that's the important thing that resveratrol is doing. So in this experiment, what we did was expose the cells to resveratrol and use an RNA knockdown approach to prevent resveratrol from upregulating manganese sod. So in other words, normally you give the cell resveratrol, they make more manganese sod, everything slows down because of this signaling pathway, we're pretty sure. In this case, you give them resveratrol, they're trying to upregulate manganese sod and slow them down, but you prevent that. And it's a straightforward way to do that experiment with interfering RNA. So when the transcript's made, it gets destroyed and the upregulation never happens. So this is just a control. SI is silencing RNA, or small interfering RNA. And what we see then, so the control is the experiment done, everything the same except we haven't actually targeted manganese sod. Here's the slowing that occurs. Here's the increased manganese sod level that occurs. In this set of bars now, we have actually targeted manganese sod, preventing it from being upregulated, and so we're down here. No resveratrol plus resveratrol, and really no big induction like what we see here. So we've achieved that, and what's shown on this panel is population doubling time again, how fast the cells are growing. And here is our control, the open bar, and here is the cells that now, again, are exposed to resveratrol. Same experiment as before, we've done so many times at this point, and normally this bar would be up here, and it's not. So the cells still see resveratrol, but the only difference is they're not able to induce this manganese sod, and there's no effect on growth. So an experiment like this allows us to actually pinpoint, yes, that manganese sod induction is a critical event for slowing replicative growth. Okay, so we've pinpointed manganese sod. That's important because, again, we think knowing the individual proteins that are being affected to modify the behavior of these cells is going to allow us to do some things. Eventually it's going to allow us to actually find other molecules that can fit into and toggle that pathway and give us the effects that we want. So what we wondered at that point was, we didn't think that the resveratrol effect on manganese sod was a direct effect. So more of that protein was being made that suggests it's a gene, it's a transcriptional effect. The gene's being used more. And so something upstream of that is regulating, and our question was, what is regulating? Now, we knew a few things from the literature. We know that transresveratrol, the active form, looks a lot like, you may not agree, but those structures are reasonably similar, resveratrol and estradiol, one of the major estrogen molecules. And there's an existing literature about the idea that maybe resveratrol was a phytoestrogen. Although, over time, resveratrol has become implicated as being a lot of other things, sirtuin modulators, cyclic AMP modulators. Stories have really, many different divergent stories have developed around resveratrol. We decided, we investigated those, and I'm not going to show you those data. I'll show you this data because it's the one that seems to explain our results. So similar structure to estrogen, and in fact, if you go in silico and you use a computer to model docking between the known structure of the estrogen receptor beta and the actual ligand, resveratrol, The computer considers it a good molecule to bind to that receptor. It will do that equally well almost as estrogen. Okay, estrogen receptor beta. I better tell you what estrogen receptor beta is. Estrogen, everybody knows about estrogens as the reproductive physiology, the role in reproductive physiology in females. That's huge. It's important. About 50 years ago, the receptor that's involved in that was discovered. At that time, it was called estrogen receptor. We now call it estrogen receptor alpha because a second version of that receptor has been identified, and that's the beta. There's actually more than that we know of now too, but at least we'll focus on estrogen receptor alpha and beta. And I'm going to tell you that I think resveratrol is working through estrogen receptor beta and not alpha. A few reasons for that, and not the least of which is that many things that work through estrogen receptor beta actually are or anti-proliferative, will slow cell growth, not just resveratrol. I'll show you those results in a minute. So the thing about ER beta is it doesn't have that major role in female reproductive physiology. The receptor itself is in places that really don't have, you'll see that receptor show up in cells that are not directly involved in female reproductive physiology. It's in lots of the parts of the brain, highly expressed in brain. It is in throughout the gastrointestinal tract. It's actually in males, it's in prostate. It's lots of places, really, in the body that don't have anything to do with reproductive physiology. It has important roles in those places, but those roles seem to be related to regulating proliferation and there's a stress resistance role there too. So ER beta, things that productively bind to ER beta generally are anti-proliferative. They will slow cell growth. This is what we saw with our resveratrol. So what we did then was to we hypothesized that resveratrol was actually exerting that effect on manganese sod via this receptor, the estrogen receptor beta. That's a somewhat straightforward experiment to do, and we've done it in a number of different ways. Here I'll show you the pharmacological approach. We've also used cells that do not have estrogen receptor beta, so ER beta knockout cells. We see the same thing. I just happen to have this graph more handy because it was published last year. So what do we see? If we do, again, the same kind of experiment where we take our control cells and we look at, here we're looking at manganese sod expression. And then we give them resveratrol, and here's a manganese sod induction. Now the extent of induction is different in the different graphs, and I'm not telling you, but it's different. Sometimes it's slightly different concentration, sometimes a different time. It's not critical to what we're talking about. What's happening in this third bar is that the cells were given resveratrol, but they were also given an estrogen receptor beta antagonist. So something that interfered with the ability of resveratrol to actually productively bind ER beta. And we see no effect. So this was the first kind of evidence we had that, at least for this effect that we're interested in, manganese sod induction and slowing in a variety of cell types, it does require that ER beta. And here's another kind of experiment that Ellen did that also reinforces that thinking of ours. Now, in this experiment, we've got the same control and then two other bars here. And what we've done is, rather than expose the cells to resveratrol, we have used either a direct estrogen receptor beta agonist, so DPN, diaryl propionitrile, which productively binds ER beta and gives you those same kinds of effects, or PPT, which doesn't bind ER beta very well, but does bind ER alpha. And what we see is we cannot reproduce any effects in terms of slowing growth or inducing manganese sod with the ER alpha agonist, PPT, nothing happens. But with the ER beta agonist, we see it. So we have many different lines of evidence to tell us that we can reproduce resveratrol's effects with DPN, the ER beta agonist. We can abolish it with a pharmacological inhibitor. We can abolish it in the knockout, the ER beta knockout. So we know that that this is the key upstream event. Okay, now at that point, Ellen, who was doing this work, Ellen Robb, became increasingly busy because we went in a couple of directions. We're still working on trying to further characterize what's going on in a regulatory way, but I'm not going to tell you about that. I'm going to tell you about something different and equally interesting, and that is, once we know that it's ER beta that is mediating this effect, now again we have a target. ER beta is a protein, so we have a protein target that we can start to think about how else can we modulate it. And again, we can go back to the in silico approach and get a computer to model this, and we'll say to the computer, I'll give you this structure and a bunch of other structures, and you tell me whether they're likely to bind ER beta in a way that gives us, allows ER beta to be active. The computer gives us some answers, and then we have to go and check them. Turns out some of the answers are correct and some are not correct. So we were interested, first of all, in some of these other things that turn up in red wine, turn up in grapes and grape leaves, the Passeid and the pterostilbene. They have very similar structures to resveratrol. And we think the important part of the structure is here, top right, hydroxyl group, and the general shape of this fits well into the binding pocket of ER beta. What's going on down here, bottom left, probably doesn't seem to be as important. Because we can duplicate, I'll show you that we can duplicate the effects with a variety of these different molecules, all of which show up as phytoalexins, as molecules that the grapes will be, the grapevines produce in response to stress. So we did a similar kind of experiment, now that I've shown you many times. And here, I've really just shown you two kinds of experiments, measuring manganese sod levels and looking at the population doubling times, those two things. Here we're looking at the population doubling times. And we have a couple of bars here, which are our controls. So this is sort of the basal level of population doubling. And then we have our resveratrol, so that's a higher value, which means it takes longer for the cells to double. They've slowed their proliferative growth. That's a good thing in a lot of contexts. I'm not going to talk about these, but I'll just we'll talk about them. I'm not going to talk a lot about them, but I'll tell you quickly that those are two metabolites of resveratrol that your body produces. When you eat or drink resveratrol, that's what your body makes, is some derivatives, some metabolites. And they show up in fairly high concentrations in blood, and we wondered if they had any activities, and they don't. So that's those two bars. But here are the two bars that we're really interested in, the Passeid and the pterostilbene, and guess what? I'm not showing this either, but around the same concentration, micromolar, low micromolar concentration as resveratrol, both of those molecules can give you the same effect. So the computer predicts they'll bind to ER beta, and they give you the kind of effect you would expect, you would hypothesize, if indeed they did bind to ER beta. This is the change now in manganese sod protein levels. You expect to see that concomitant with the slowed growth, manganese sod would be induced, and that is exactly what you see. So here's resveratrol, here's a bunch of controls, the ones we're not interested in right now, and Passeid and pterostilbene, which are very structurally similar to resveratrol. So, other vinifera phytoalexins are structurally similar to resveratrol. Do they have the same effects? The answer seems to be yes. Now, again, I'll qualify that by saying we've now characterized 3 of probably dozens, anyway, of structurally related molecules that you would find in red wine and in grapes and in leaves. It'd be very nice to look at all of them to really decode the red wine. And that's just a matter of, at this point, it's been a matter of what's actually commercially available. And a lot of those are not. We're trying to set up collaborations where we can get those molecules from other researchers, because that's the level at which they are available. Oh, right. Just to show you quickly, we can make those same effects of the pterostilbene or the Passeid go away if we don't allow the cells to upregulate manganese sod. So we think they're working through ER beta, and we've shown that. And we think that it requires this manganese sod induction. We showed that for resveratrol. Here it's being shown for the pterostilbene, at least. Now, you'll see cholesterol, genistein, diadzine, a whole bunch of other molecules that I haven't mentioned, but we're working on so some of these are not produced by red wines, and that's why I haven't mentioned them. But just as red wines produce those, just as the grapes produce phytoalexins, all plants are doing this, and many of them are giving similar effects. I'm going to brush by that, and cut to the chase. If you prevent the manganese sod induction for any of those things that are ER beta binders produced by plants, you don't get the effect on growth. You don't get the induction of this manganese sod, so that just goes away. So again, it comes back to, we think we can now find a whole bunch of different molecules that bind ER beta, or are agonists of ER beta, and that's communicated into the cell as a turning on manganese sod, and that ultimately is communicated as slowing cell replicative growth. So I promised to talk about cancer. What I've been talking about up until now is actually almost all data from normal cells, normal human cells, or at least mammalian cells. So we see it in it. It doesn't matter what species. Those anti-replicative activities are interesting in normal cells. There's all kinds of reasons for that have to do with aging and the maintenance of replicative capacity in mitotic cells in vivo. But there's a specific application that's also interesting, maybe more interesting, which is that resveratrol has been implicated as an anti-cancer, and many of the red wine molecules actually are in this category. And so what we've tried to do is just understand whether that effect, that anti-cancer effect, is actually the same thing we have already seen. So anti-proliferative effects in cancerous cells, whether they're working through the same pathways as what we saw in normal, not cancerous cells. Now, interesting literature in the literature on cancer. This is old, so it's a little fuzzy. But in the early 1990s, late 1980s, a group in the United States started to publish work showing that a lot of cancers, a lot of cancerous cell lines express very low levels of this enzyme, manganese sod. So cancerous cell lines, they're modified in a number of different ways. It's not necessarily the case that this is the only thing going on, but this is one of the things that can be identified as being changed in those cells. So this, we've got manganese sod activity and the transcript level, mRNA level. This is a normal cell. That's the same matching cell, but this one has been transformed, is now cancerous. Normal, transformed, and cancerous. So it's something that shows up regularly. I didn't put a number on it, but I would estimate, well, over 50%, to be conservative, of cancers show this phenotype of low manganese sod expression. Prostate cancer in specific shows that phenotype, and that, you'll see why that's interesting, because I'm going to tell you a little bit about prostate cancer in a couple of seconds here. But prostate cancers, if we look at the normal levels of manganese sod in the prostate epithelium versus the levels of that enzyme in a cancerous prostate cell, we can see that it's way down. This is an activity gel, same kind of thing. General point is that in these cancerous prostate cells, manganese sod expression has been depressed. We think that's important because we know that if you were to do that in and of itself, it would speed up replicative growth rate. Here's a really important experiment done by someone else and published in Oncogene a few years ago. Now here, they're using a transgenic approach. So they're putting manganese sod back in. So we recognize that in the cancerous prostate, manganese sod levels are low. The cancer cells are growing rapidly. That's actually shown here. This is the rate of growth of these prostate tumors. So these are days and the size of the tumor. These last three lines where things are much more static, the tumor has not grown nearly as large. are overexpressing, or actually they have been rescued. The low manganese SOD expression has actually been rescued by putting in the transgene, some extra copies of manganese SOD, and forcing its expression. Now they have to make the protein, even though something had happened to reduce the level before. That was enough to slow them down. Another little bit of information is that some cancers express ER-beta. There are cancers of the GI tract, some breast cancers even. In breast cancer, it's a good prognostic indicator. Having a lot of ER-beta in a breast cancer tissue, cancerous breast tissue, is actually a good thing, because that can be targeted for anti-proliferative effects. Prostate cancers are one example of that. They are express particularly high levels, many of them, of this ER-beta. So that's shown in this table, but for the sake of time, I'm going to go through that very quickly. So just to summarize, linking this together, many cancers are characterized by this reduced manganese SOD expression. And if you fix just that one problem, you can slow them down. You won't stop them from being cancerous cells, but you will slow down the rate at which they grow. That's arguably just as important, because it's the size of the tumor that matters ultimately, and if it grows very slowly, it may not be a problem over a lifespan. We know that ER-beta stimulates manganese SOD expression. We know that prostate cancer cells have ER-beta. And we know that these things that the grapevine produces are ER-beta agonists. The question is then, can we use those things that grapes and grapevines produce to actually slow the rate at which prostate cancer cells grow? So that became our specific question. Just recently, and this was work done by Ellen, but also an undergraduate named Devin Payne in the lab. So we did the same kinds of experiments that we've now done so many times in the normal human cells. What we did is to just repeat them in these cells that were actually isolated from cancerous prostates, human cancerous prostates. And we measured the same output, population doubling time, and we were looking at our known agonists of ER-beta that we'd identified previously. These are the things that show up in red wine and other grapevine products, Resveratrol, and all of the molecules that look like Resveratrol, pterostilbene, Pasaids, and again, we have not been able to do some of the others that we think are highly likely to be effective just because of lack of availability. Here are some controls, some negative controls where nothing's added. DMSO here is just dimethyl sulfoxide. It's the vehicle that these things are added in, because they're not very soluble, so this helps them increase their solubility. No effects. DPN, again, is our strong, directed ER-beta agonist, and this is a pharmacological. This is something that was invented in somebody's lab. So it is owned, it's an owned molecule, as opposed to the phytoestrogens, which are not. And then increasing amounts of resveratrol, and we see increasing effects in terms of slow growth. And one thing I'll just point out really briefly is that resveratrol is actually particularly good at slowing the growth of prostate cancer cells. It'll do that with any type of cell, but it tends to really work well on this type of cancer cells, and we think that it's going to work really well on any type of cancer that expresses lots of ER-beta. We think that's why it works so well, is these prostate cancer cells, for whatever reason, express high levels of ER-beta. And this last one is pterostilbene. So 10 micromolar pterostilbene, and here's 25 micromolar resveratrol. If we drew a line across here, we would see that there actually you can get a very similar effect at 10 micromolar pterostilbene as you can with 25 micromolar resveratrol. So it's an effective substitute for resveratrol. So again, we think that probably all of those molecules are going to give us the effects we want. Here's our manganese SOD. This is the thing that we think is controlling everything. And again, we do see that it's being induced by what's going on here. Now, there are no error bars, and partly that's because we just haven't done the stats on these yet. It's actually, this is the most recent data that we have, or at least I haven't been given the stats on these yet, if they haven't done. I can tell you that this is going to be a real difference. And actually a number of different people have done similar experiments showing the same results, so I'm confident that this is in fact a real difference. So what we see then is an induction of manganese sod and a slowing of growth in specifically these prostate cancer cells. So we can conclude that at least 2 of those phytoalexins produced by stress vitis vinifera will give us that effect, which is a really important effect. That's a big change in the rate at which prostate cancer cells grow. If you could reproduce that in vivo, the rate at which a tumor coming from those cells grew would be substantially reduced. And you have to remember that it's an exponential thing. So when you slow the rate down, 1 cell becoming 2, becoming 4 and 8, all of that, you're staying way down in the range of this exponential function. So what are we going to do next with that information? We've convinced ourselves, almost, we have some more experiments to do with the prostate cancer cells, but we really would like to see, because we can go out and actually in the literature we can find types of cancers that express a lot of ER beta. We have that information. We can get those types of cancers and repeat this work, which is one of the things that we intend to do. Some people in the audience are going to do this work. And we can also, we're very interested, particularly interested in looking at other viniferins. And then finally, we need to always, in the lab, when we find these things in cell culture, we always try to move them into a mouse model. We'd like to know that they work in vivo. We can't do that, but we can put these things into mice and characterize their ability to do the same thing as they're doing in a dish inside of the live animal. So can they slow prostate cancer growth in mice? Okay, so general conclusions and prospectus. So we know that the ER beta agonists are stimulators of manganese sod, which is a known tumor suppressor enzyme that slows growth. We've shown this for a couple of the viniferins. The 2 things that we think are linked, manganese sod induction and slow growth. And where do we go from here? We need to build on and expand that story. And all of that work that I showed you was sponsored by a variety of funding agencies. Primarily this has been by NSERC via the Vanier Scholarship to Ellen Robb, and also the operating grants, but also the CFI and Ontario government for the infrastructure that we use, the equipment that we use to do the work. And that's it. Thank you very much.
Thanks, Jeff. That was such an exciting talk. I'm just so excited about this information, especially since we're in the heart of grape and wine country. Being cool climate, of course, we have these fungal disease pressures. We know already that our grapes tend to have higher levels of some of these compounds, so it'll just be fascinating to follow this story to see how it progresses. We've got time for some talks or questions, so please, are there any from the audience? Blind Mr. Early in the talk, you were showing some results from your mouse model. When you delivered resveratrol in a high-fat diet, it had quite a significant impact, but not just from a regular diet. Are you thinking along the lines that, possibly because of its hydrophobic characteristic, that red wine with its ethanol content, might it also be a good delivery vehicle for getting these types of compounds into a system? Is that a possibility, or is that requiring much more work too?
So the question is, does the vehicle matter? Going back to one of the earliest slides, the way resveratrol is given to mice influences the size of any effect that you would observe. We've seen that. Other people have seen that. Not measuring, they've been measuring something different, but it is known that the vehicle matters, how resveratrol is delivered. And we know that a high-fat diet is where you'll really see the effects of resveratrol. Now, why is that? We do, in fact, think that it's because of the packaging, in terms of fat being surrounded and perhaps even protected by, certainly solubilized in fat in the digestive tract, might be really important in terms of promoting the uptake and protection of resveratrol in the body. That's a possibility because it is rapidly metabolized. And it might be that is slowed down by being packaged in a fatty meal. Real possibility. And yes, the other thing is, can you deliver it in wine? Now, those experiments, they could be done in mice, but probably I would not do them in mice. I think that those would be great experiments to do in people. Gary Pickering, of course, has got a red wine. He's done some experiments with red wine that's been supplemented with resveratrol at pretty high levels. It tastes fine, apparently. And so that is a way to do a study like that. Now you have to have people that were willing to give you some kind of sample downstream. In immune cells, you could measure some of these things in immune cells as well. So it could be done. We do think the vehicle matters a lot. And there are a number of other people. We've experimented with Paul Zalisco packaging the resveratrol in silicon, silicon nano encapsulation. Other people have taken other approaches. Some of it is showing signs that it might work.
One other question from me before I go back to the audience. Just in terms of these other phytoalexins that are in grape and Vitis vinifera, approximately how many of these compounds were there? That one table, like 7, 8, 10?
So I think that table is a subset of all the ones that I have read about. Seems to be, I mean, some people in the audience might know more about this, but I have the impression that just Vitis vinifera might produce more than a dozen, anyway. And then I haven't mentioned other plants that produce their own versions of these, many of which give similar effects. So some of them I included, we've worked with some of them, and I included some of those results but brushed over them. So we're talking about a lot of molecules. If we expanded beyond grapes, then we would be talking dozens, several dozens at least. Yeah. And some of them, again, we can feed them into a computer program that would simulate their ability to bind ER beta. It predicts that a lot of them would actually do a pretty good job of that. But ultimately, we have to characterize them in a cell system, an intact cell. And we're doing that. Ellen has screened maybe 10 to 15 of them. And of those 10 to 15, I think 7 showed signs that they were working that way.
Any other questions from the audience? Any from the online guests? No? Quiet group today? Well, Jeff, I'd like...
Actually, a general question. Maybe it's a novice question. But in regards to the red wine and white, is the same anti-cancer fighting cells within the grape?
Yeah, that's a great question. And I'm probably not the best person to answer this because I'm not a wine person, but my understanding is that when you make red wine, the wine is being fermented on the skin, and you've got a lot of these on the skin. They need to be on the skin because they've got to be actually on the outside of the skin where they can interact with any fungi that are doing their thing and slow down their growth. And so when you're fermenting wine and the skin is present, and those molecules like to be in an ethanol solvent much more than they want to be in water, you can have some extraction. And I think the white wines are not fermented that way. The skin is not present. And so you don't get the ability to capture those in a forming white wine. So when you measure those things, you see higher levels in red wines. Those things being the phytoalexins I mentioned, you see higher levels in red than in white.
Okay, well, please join me in thanking Jeffrey for a great talk this afternoon. We're going to talk about our first edition. And just a reminder to the group that our very last COVI lecture is happening next Wednesday, and it's by one of our fellows, Dr. Don Sear from the business faculty, and the title is A Cost-Benefit Analysis of Entering and Exiting Vineyard Operations in Niagara. So hope to see you next week. Thank you.